then you can proceed lah. All right.
Yeah.
Okay. Okay, we are live.
Hello, Marie. Good afternoon.
Shamsun, maybe you can start.
Yes, sure.
Yes, let's start it now.
Good afternoon. Welcome to the invited lecture series A of ASOMPS. And this is transmitted from Putrajaya, Malaysia, but definitely we are from different time region. Our first speaker is very honorable Professor Mary Granson. It's indeed an honor to introduce the eminent scientist in the field of natural product chemistry, Professor Mary Granson. Professor Granson did her PhD from the University of Cambridge in 1977, and then followed by postdoctoral research as a fellow of the Royal Society in Rome, Italy, from 1977 to 1978. She served as a research fellow in Cambridge as well. And there are a lot of activities and there are a lot of achievement by Professor Granson. And Professor Mary Granson is now Emeritus Professor, School of Chemistry and Molecular Biosciences, the University of Queensland. Professor Granson made a Member of Order of Australia in 2019 for her extensive contribution to the education. She is an elected member of IUPAC Bureau, and she has participated and collaborated several hundreds of projects in Southeast Asia. So we are welcoming Professor Mary Granson for her eminent lecture at this invited Series A. Thank you.
Okay, thank you very much, Professor Shamsun, for that very generous introduction. Before I share my screen, hopefully somebody will tell me if they can't hear me properly. But before I share my screen, I just wanted to say, first of all, thank you very much for the wonderful opportunity to talk at ASOMPS. And I think I'm also going to comment that although I'm technically retired, I'm not retired, right? I'm active and energetic, and my research is going to continue. And I'm also going to brag a little bit. Last week, I was in an election for the position of incoming Vice President of IUPAC, the International Union of Pure and Applied Chemistry. And I'm sad to say I lost the election by one vote, by 69 votes to 70. So I'm honored to think that there is value in people like me standing for office. And the main reason I mention it is because I'm going to have another go. And I'd like all the young people in the audience to know that when you put your head up above the parapet and go for leadership, it's something to celebrate. And you should always be prepared to lose, but just give it another go. So watch this space in 2 years' time. Okay, so I'm going to put IUPAC to one side, and I'm going to share my screen. This is the bit I'm terrified at. If it doesn't, there we go. All right. Silence. Wow. Can everybody see my screen?
Yes, it's visible.
You see the talk?
Beautiful.
Right. Okay, thank you so much, everybody. Now I've just got to put this on slideshow mode. I'm at home struggling with several monitors. There we go. There we go. All right, all organized. Right. So we're going to talk about mollusks. And I think I'm going to move to the next picture straight away. Just want to move that out of the way. Oh, let's go back. All right. So the senior people in the audience know that I worked for many years on marine sponges. Nudibranchs, these little colourful mollusks that we can see in this picture, eat marine sponges. And so there's a link between the chemistry of the sponge and also the mollusk. And many of the mollusks that we work on are quite small. This one is about 2 centimetres in length. We work on individual specimens. So the extract sizes are very small, 5 to 20 milligrams. The actual organic chemistry, the natural products chemistry is quite traditional. We're working with sub-milligram quantities of metabolites, flash chromatography, HPLC. And this talk is going to be about the 2D NMR and the computational work that we do to evaluate not really the planar structure, but the 3-dimensional stereo structure. Now, before I move on, I need to tell you that in this talk, I'm not going to mention at all our ecological and biological work, which is going to continue in the next few years. All of the work today was covered by my final chemistry PhD student, a wonderful lady called Louise Forster. And in addition to doing the chemistry, Louise has spent quite a bit of time dissecting the mollusks, so separating out the intestinal tissue from the colourful mantle here, and in particular, the mantle rim, this little coloured edge. And so what we're often doing is comparing the chemistry of the intestines with the chemistry of the mantle tissue. And the reason for that is that from an ecological point of view, when an animal is attacked, it folds its mantle out over its body. And so it's the mantle tissue that the animal needs to protect. And so it sequesters, the hypothesis is that the mollusk sequesters certain selected potent compounds from its sponge diet and transfers them by mechanisms as yet unknown to the mantle rim to protect the animal. So that's going to...
Sorry to disrupt you. Can you just angle your camera so we can see your face?
Oh, I know. I see what you mean. You mean that,
yes,
don't you?
Yeah. Okay, now we can see you clearly. Thank you. Sorry.
Thank you. You don't really need to see me, actually. Everybody, I think most people know me. Sorry, I moved that because the laptop, the back of the laptop is actually partially hiding the screen, but is that okay?
Yeah, it's splendid.
That's good.
Thank you.
Okay, all right. Let's move on to the... come down here. Do it that way. All right, okay. Now to illustrate the difference in chemistry between the mantle tissue and the intestines of the animal, here's a study that we published in about 2016. So what you can see in the circle are 5 different species of chromadorid mollusk. And to our eyes, they clearly all look different to each other. But in fact, to a fish predator, these 5 species would look quite similar. The yellow patterning around the edge of the mollusk would be the only way that the fish predator has of distinguishing these different species. Around the outside of this diagram is the typical chemistry that we see for the gut tissue of these animals. And you'll see that each individual species has different chemistry. We've got examples. Compound 2 is a miroterpenoid, 3 and 4 are sesquiterpenes, 6 and 7 are diterpenes, and compound 5 is a sesquiterpene. What these 5 different species all have in common is the presence in their mantle rim tissue only of the potent cytotoxin macrolide compound called latrunculin. And so this is the defensive compound. So I think this illustrates nicely the point that you actually have to consider the gut tissue separately to the mantle rim tissue, because if you extracted the whole animal and didn't separate these 2 tissues and did some ecological experiments, you might infer the wrong conclusion as to which chemical present in the chemical extract is actually the defensive compound protecting the animal. So that work was published back in 2016. I want to move on now to Louise's work. And this is just a little appetizer. This is from a species, Goniobranchus splendidus, and you can see that in the top left-hand corner. And Louise isolated and characterized over 20 different compounds from the extract of a single animal. And I'm showing you here a compound goniolene, which has got a cyclohexanol structure. It's got a trimethyl substituted cyclohexene, and then it's got this dioxide bicyclo system there. We're going to see this motif coming up through much of the talk. Underneath it is a different compound, a different carbon skeleton. It's got that highly oxygenated component, but it's kind of got an extra carbon sticking off here. It's an epoxide, and it has still that trimethyl cyclohexane substituted moiety. So there's a lot of stereochemical elements here, and we deduced these structures and 3D stereochemistry of these metabolites, of course, by 2D and also by NOESY spectra, but we informed ourselves about the likely stereochemistry by doing some computational work. We used a computational method called DP4+, which is after you've modeled candidate diastereomers and identified for each diastereomer a number of possible conformers, three-dimensional shapes, the theoretical calculations allow you to predict which one of those candidate stereoisomers matches best to the NMR data, to the proton and to the C13 NMR data. And so it was on this basis of this theoretical work that we deduced that the likely stereochemistry of the new epoxygonolide is as shown in the bottom structure. And we reinforced this conclusion by thinking about the biosynthesis of the compounds. This is a very busy slide. Starting from it's a diterpene compound. So starting from the well understood diterpene precursor for this group of marine metabolites, and we refer to this as a spongian skeleton. There's quite an extensive rearrangement which removes this carbon, breaks that carbon-carbon bond, and critically, this methyl group migrates to that position. And this generates the structure shown here, number 1. And of course, various oxidation processes take place. And we speculate that to get to the modified diterpenes, goniolene, and the epoxygoniolides, that you have to close up by a carbanion attack on an epoxide. That's the epoxidation of that terminal double bond. And that puts in place the cyclohexanol ring that I identified in goniolene. And what we were really interested in was how this would relate to the epoxy compounds. And we recognized that a so-called Grob fragmentation, which involves breaking that carbon-carbon bond, effectively opens that 6-membered ring. It generates an alkene and an aldehyde. Epoxidation of that then leads us, after we've sort of closed here to make the lactone, leads us to the 2 epoxides. So we were utilizing in our thinking about the stereochemistry, the relative configuration, and in terms of the absolute configuration, working from the generally understood absolute configuration for these diterpenes, which is shown there, that led us conclusively to the bottom 2 structures. To confirm those structures, it would be really nice if at some point in the future, somebody did some chemical synthesis of them, because they're engaging candidates. All right, so now I want to move on to the main part of today's talk. And this is Goniobranchus koi. What I show on this slide are the known compounds that Louise isolated from the animal. And I've divided them into 3 different groups, pink, yellow, and green, just to help us out. So this is a perhydroindane skeleton. And notice we've got that oxygenated motif. The yellow structures are a hydronaphthalene moiety. Once again, we've got that dioxo system and an alternative rearrangement of the dioxobicyclo system. Now, today's talk is going to concentrate on the bottom green skeleton, which is a perhydroazylene system. And we're going to focus on some derivatives of dendrolylide A. But I just take this point here to highlight the wonderful set of compounds that Louise isolated and characterized rigorously. Right, so dendrolylide A is a very well-known compound. It was first isolated from a marine sponge. Notice 22 carbons, 5 oxygens. We pay attention to that mass spec data. And we have that exomethylene functionality at carbon 10 that I've put a box around. The compound shows anti-inflammatory activity and is believed to be a predator deterrent, but the ecological evidence in support of that is not terribly rigorous. And I'm going to refer to this skeleton as the dendrolane scaffold. And the other piece of introductory information you need to know is that we need to recognize the various conformers, the three-dimensional shapes possible for a 7-membered cycloheptane ring. As shown here, a chair, a boat, a twist chair, and a twist boat. And for cycloheptane, generally the major conformation, the energetically favorable conformation, is the twist chair one shown here. Once you put a double bond into the system or some rigid structure, and in our case, it's going to be an epoxide ring, we're going to discover that the chair form becomes energetically favorable over a twist chair. All right, so moving on rapidly to the first molecule that Louise isolated. Here's our dendrolane skeleton, and we have an epoxide group across that bridgehead position. Here we've got the proton signals for the exomethylene. And the bottom spectrum, the blue one, is dendrolylide A. And in the epoxide derivative, in the red spectrum, those signals seem to be missing. And another of other signals in the spectrum are unusually broad. It was quite clear that this was a derivative of dendrolylide, and the extra oxygen told us that we had to position either a hydroxy group or something like an epoxide group in the molecule. Now, the spectrum I'm showing you here is at room temperature. And we began to wonder if the fact that signals were broad or signals seemed to be missing, whether there was some conformational averaging going on, so that on the NMR timeframe, because of the fast interconversion, if there's a slow interconversion, we will see different conformers. But if there's fast interconversion, we're going to end up with fuzzy images effectively in our NMR spectrum. So the first thing we did was to get a higher temperature and notice that a broad signal suddenly starts to sharpen and we get a bit more structure here. So immediately we knew that we were indeed seeing conformational averaging. I've highlighted in pink here the proton signals that were affected by line broadening or loss of structure. So these effects were evident both in the carbon spectrum, but in the proton spectrum, but also in the carbon spectrum. So in order to see all of the protonated carbon signals, when we were running the HSQC spectrum, we had to run that at 45 degrees as well. At 25 degrees, some of the correlations were missing, and it wasn't possible to validate the full structure. Coming back to the proton spectrum, I've told you so far about what happens if you warm the sample up, but much more interesting was what happens if you cool the sample down. And we ran these experiments in deuterochloroform, and as we went down to -5 and then to -50 degrees, you'll see a second set of signals appearing here. And of course, we see better compared to the red spectrum, we see a doublet signal rather than just a broad lumpy signal. So again, this is clearly conformational averaging, and at low temperature, what we're doing is effectively slowing down the rate of interconversion of the conformers. And in other words, we're beginning to see individual signals for the individual conformers. It's possible to do some calculations and we calculated that the activation barrier to the conformational change was about 15 kcals per mole. And the question was whether that was something to do with the conformational change in the 7-membered ring. By the way, the 5-membered ring adopts an envelope shape. Or alternatively, we had to consider the fact that the carbon-carbon bond joining the two halves of the molecule together is highly substituted. It's in fact pentasubstituted. So the other conformational possibility was that we had restricted rotation about that bond. So what we had to do next was to do some modeling. Oh, by the way, just in case you want to get some background on how we calculated that activation barrier, we did some arithmetic. It's a very classical derivation of the A-ring equation, and it simply requires you to know the coalescence temperature and the difference in frequencies for the non-equivalent protons. If anybody's curious about how to do that, please flick me a little email and I can explore it with you. All right, let's move on quickly. We had to do some molecular modeling, macro model, and we got 8 individual conformers that were of reasonable energy. And then we did density functional theory to reduce the number of conformers to a manageable number that were energetically favorable. So within about 0.6 kcals per mole of the lowest energy for the major conformer. And we found there were 3 types of conformers, and they differed in conformation for that 7-membered ring. The major conformer is the one shown in green, and that's a twist chair. And there were 2 other minor conformers, one shown in blue and the other one shown in pink, which corresponded to a chair or an inverted chair. So the interchange that we're seeing here between the conformers is from a twist chair to a chair form. And so it's no surprise that it's the proton signals in the vicinity of that 7-membered ring which are being affected. Now, what we didn't know at this point was the configuration of the epoxide group. And we cheated a little bit, and of course it solved the structure for us. Louise, who's brilliant in the lab, crystallized the sample, which had 1.4 milligrams, and the X-ray structure was solved for us on the synchrotron by our colleague Jack Clegg. And what was really quite interesting at this point was to discover that the conformation in the solid state was quite similar to that minor conformer, the pink conformer, where we've got a chair form for the 7-membered ring. All right. Now, for comparison, we did some molecular modeling and conformational analysis of the parent dendrolylide A skeleton, for which we had never seen conformational averaging in the spectra. We had never seen line broadening effects. When we did our macro model conformational searches, we found 4 conformers. And then using the DFT theory, we reduced it to 2. So 2, essentially only 2 conformers that had to be considered. And the picture's quite different. These 2 conformers have a very similar 3-dimensional shape for the perhydroacetylene, and they differ in the arrangement of the right-hand hemisphere, the dioxobicyclosystem, relative to the left-hand hemisphere. So you can see the blue and the green here, they are quite close to 180 degrees apart. So these 2 conformers differ by rotation about that congested bond, but nevertheless, there must be reasonably rapid rotation about that bond because we don't see line broadening effects in dendrolylide. These calculations and modeling were done for us by our colleague, Greg Pierens. If you want a bit more technical detail on this, please do look at our recent publication. All right, so I'm going to move on very rapidly because I know I'm about to run out of time. The second compound I just want to tell you about very briefly, instead of the exomethylene group, has a keto group here. So it's a NOR compound. And the molecular formula told us immediately that we were missing a carbon and we had added an oxygen. And of course, we got a ketone signal in the C13 NMR. And critically, the signals for the exomethylene group were missing in the proton spectrum. And once again, we saw some line broadening effects in the spectra. So in the carbon spectrum run at room temperature, not all of the carbons were present. Of course, this is a 2D spectrum. And so the spectra had to be run at higher temperature at 45 degrees C. We did once again some temperature dependent experiments. Here I'm simply showing you what happens if you go up in temperature. And so a signal that's quite broad and fuzzy at room temperature sharpens as we go up to 45 degrees C in chloroform. By the way, you can also run these temperature dependent experiments in acetone and you can get down to lower temperatures. We got down to about 190 for the first compound, the epoxide. And at that temperature, we actually saw 3 sets of signals, not 2 sets of signals. All right, so we once again did some molecular modeling for the this keto compound. Again, the conformational change is in the perhydroazylene ring system, but there was also some movement, some changes in the conformation in the shape of the right-hand dioxybicyclo moiety. And I'd simply refer you to our publication for the technical details. All right, now just the final compounds that Louise isolated, and these are my last 2 technical slides, where she isolated the epoxide here of dendrolylide. And so instead of previously I was talking to you about an epoxide across the bridgehead position, here the exomethylene has been converted to an epoxide. And it's not 100% clear that this is a genuine natural product. It could in fact be an artifact formed by oxidation of the exomethylene group during isolation, but it's quite well documented that those sorts of systems are quite slow to oxidize. Nevertheless, we had to deduce the configuration at the C10 position there, whether it was an R configuration or an S configuration. We did some nosy work and modeling and DFT calculations, which I won't go into in detail, supported a 10R configuration. And we took a sample of the parent dendrolylide A with the exomethylene group and treated it with the classical reagent for doing epoxidation, metachlorobenzoic acid, and we actually obtained both diastereomers. Now, the final point I want to make is that we also isolated 2 aldehyde products. And I think it's best to explain that if I move to the next slide here. Here's our major epoxide that we isolated. In the crude NMR spectra of the extract, we saw evidence for a second epoxide, this minor epoxide, which necessarily had to differ in configuration at that C10 position. And we deduced that it was unstable and rearranged quite rapidly to an aldehyde product, which I'm going to call the major aldehyde, which has got the stereochemistry shown. And we also, because we had isolated samples of the major epoxide, were able to evaluate that over time, its NMR spectrum changed and the epoxide signals disappeared. And we saw signals for an aldehyde product, which has got this configuration. And so what we're seeing here is an example of the classical Haus-Beinwold rearrangement, which is essentially a 1,2 hydride shift. And in terms of the stereochemistry, the hydrogen that moves is anti-periplanar to the carbon-oxygen bond that's breaking. So in this way, we were able to relate the configuration of an epoxide with its aldehyde product. We haven't published that little part of the work. That's something to do later this year. In this final technical slide, I'm simply reminding you about how these compounds might be generated from that diterpene skeleton, break this carbon-carbon bond here, and then how do we convert our 6,6 ring system to the 7,5 perhydroazylene?
Sir, grandson, it's already 25 minutes.
Yeah, I'm on my last technical slide. Thank you. Thank you very much for that reminder.
Thank
so this
you.
rearrangement generates the dendrolane skeleton and then oxidation, and we close to make that bicyclo system. So I just want to thank Louise there for doing the work, and Greg, who did the modeling. Karen is my biology collaborator, responsible for all of the collections. I very much want to thank you all for listening, and I'm just going to put up a flyer for the Global Women's Breakfast in 2022. This is an initiative that I work on behalf of IUPAC, and in 2021, even though these breakfast events were virtual, we reached women chemists and also men chemists in 70 different countries. And Malaysia was one of the participants, as well as Indonesia. So if you're interested to know more about that, please get in touch with me. I'm going to leave that one up. Thank you so much for listening to me. I'll pass it back to the moderators at this stage.
Thank you, Professor Mary Garson, for your very insightful and interesting lecture. So for participants, you can actually type in your questions at the Slido chat box, and we can read out the questions for you for the presenters to answer. Okay, so far I'm not able, or there are no questions posted yet, but I have a couple from me.
Okay.
Okay, I'm actually interested with the molecular modeling tool, DP4+. So is that software that has been developed by your own group?
No. That's the actual DP4 programs are developed by groups in the UK and groups. There's also a group in Argentina and a group in the States. There are a number of versions of it, DP4, DP4+. So essentially what it is a sort of statistical approach. It's simpler than doing full-blooded density functional calculations. What you do is you design candidate diastereomers, and the program calculates the probability, the chances that a particular diastereomer fits well to the NMR data. And generally, if a fit is greater than 95%, then it's considered to be the correct diastereomer. There is a very large body of literature on that. It's an important asset tool that the natural product chemist should be using to elucidate complex stereochemistry. So I'd like to encourage everybody to consider using it. It just gives you so much more confidence that you've got a structure correct. I think there's a presentation in this meeting about using theoretical methods. So you basically do some modeling of conformers and then progress to the DP calculations. For me, that's a little bit of a black box, but actually what I do as a chemist, when the modeler tells me what conformations they found, I take my NMR, I take the NOE data, and we sit there with a model, twisting the model around to see if it just sort of matches up with what we're seeing in the NOSI spectrum. So we are engaged at every point. But the actual detail of how the calculations are done is very much a black box for me.
Okay, all right, thank you.
But it's a wonderful tool, as I hope
yes,
I should.
Yes,
it's not terribly difficult, actually, for people who know what they're doing. And my collaborator, Greg Pierens, is very happy to help people. I think the original DP program was developed by Jonathan Goodman and his group in Cambridge. But there's a group in Denver, and there's a wonderful group in Argentina who work on these computational methods. So it's a rapidly moving area of natural products chemistry.
Yes.
But line it up also with biosynthesis and using just a mechanism as well, and join all of those bits together.
Yeah. Okay. So I can just allow one more question. There's a couple. Yeah. One from
Mari-san. Yoshi,
Prof. Asakawa.
I have a very very short question. How to extract your animals' molluscus with solvent or the dry material after freeze dries, or what do you?
No, we don't freeze dry them. No. We take the frozen animal, and it's a wet extraction with actually with acetone, and then partitioning into ether. Acetone extraction, because once the animal is frozen, the cell structure is broken down and the compounds can be removed quite easily. You don't have to do with plant material, you have to do like 3 days, soak it in methanol. That's not what happens here. It's actually quite quick. It's half an hour in acetone or in ether, but we don't freeze the animals. Hate to say it, occasionally we have to dissect living tissue,
Yes.
which we're very uncomfortable with.
Mm.
But from time to time, if the animal is very small, the only way to separate the rim tissue from the intestines is to do it while it's still alive. And of course, you do that under a dissecting microscope.
Okay.
And I generally leave the lab at that point.
Okay, thank you so
Thank
much.
you.
Thank you.
Thank you so kindly for listening to my lecture.
Okay, I think if with respect to time, we shall conclude here. I think if we are given the pleasure of time, so much discussion can go on, I think, with this section. Yeah, so there are some questions in the chat box now building up, but I think we shall post the questions later to the speaker. Maybe at later times it can be given feedback. So with that, I would like to thank Prof. Mary Garson for her to be able to spend her time with us live and to present us, yeah, with her fabulous works.
Thank you. Thank you so much.
Thank you very much. Yeah.
Thank you.
Over to you, Prof. Shamsun.
Thank you, Prof. Rosanna. So our next speaker, Professor Al-Fahmi, and it's indeed a great pleasure to introduce the eminent Professor Al-Fahmi of this region. He received his doctoral degree in pharmaceutical biology from University of Groningen, Rug, which is in Netherlands. In 2006, he received his PhD. And currently he is serving as an associate professor in the pharmaceutical biology department, the School of Pharmacy, Bandung Institute of Technology. He has been appointed as Vice Dean of Academic Affairs, School of Pharmacy, Bandung Institute of Technology. And he also headed the University Center for Nutraceutical Bioscience and Biotechnology Research Center, which is in the Bandung Institute of Technology. And he will be presenting his talk on production of active compounds from medicinal plants using genetic engineering techniques, status, challenges, and prospects. Please, Professor Al-Fahmi.
Okay, thank you very much, Professor Shamsun Nahar Khan, for the kind introductions. Good afternoon, everyone. Professor Othman as well. Yeah. First of all, I would like to thank the ASOMPS committee for the very grateful this meeting that who invite me to give the lecture today. So I hope that my presentation can share our research finding to you. Let me share the presentations. Could you see the presentation?
Yes, we can see.
Okay, that's good. What about my voice? Is it clear
enough?
Yes,
it's clear and loud.
It's good. Well, because of the time limit that I have, so I can start it with my presentation today. That I'm Al-Fahmi from the School of Pharmacy, Bandung Institute of Technology. Maybe for whom have never been in Bandung, this is our building, the main building. Then we just last year celebrate 100 years, 1 century, a centennial celebration. So we have 101 years now. So the title of my presentation, that's Professor Shamsun Nahar Khan already introduced to you all, that production of active compounds from medicinal plant using genetic engineering techniques. Yeah, maybe I can talk about the status of our research, also challenge and also prospect in the near future. So I would like to introduce to you about the condition now in development of herbal medicine, especially related to the secondary metabolites. That's production of secondary metabolite from plant is usually with a low level, right? It's pure. So you can imagine vincristine, for example, vinblastine from Catharanthus ratios from the flower. We only have 0.01%. So maybe you can imagine how many plants you can collect. You can get 1 kilograms pure compound. So this is much the problem. Also, pyropilotoxin is an anti-cancer agent, nice anti-cancer agent from pyropilotoxin. And I would like to tell you about our research in artemisinin. Artemisinin is antimalarial drugs from Artemisia annua. So that's, I would like to present to you that biotechnology is the method that can be used to improve the production of secondary metabolite from plants. So I think in this even I would like to focus in more focus in artemisinin production with the genetic engineering, synthetic biology, or I can say also the combinatorial biosynthesis. So actually in our lab, we have already done applied biotechnological techniques to improve the production of, maybe you know that this is the very interesting, important plant for the herbal medicine, andrographolite from Andrographis funiculata, that now in Thailand also have the national compounds for treat COVID. And we also did some research biotechnology with the lignan and phyllanthus neroli, asiaticoside from Cetulla asiatica. And then finally, I would like to more focus in these compounds, and this artemisinin. Artemisinin, you look at this compound, this complex structure. So that is the people not able to synthesize chemically. So I think, but the low content of artemisinin also forced us to find a new technology to do that one. So what the approach is to apply the biotechnological technique in the secondary metabolite production. We do in a very classical way, cell culture and medicinal plants for secondary metabolite productions, agrobacterium mediated transformation, cloning and expressions. So I would like to explain to you about the second and third ones in several compounds. So this is our nice, I like this picture. This is highly rooted culture, genetically transformed genetic culture with Agrobacterium rhizoganis. So that's mean the from Artemisia annua. In wild types, the root doesn't have any artemisinin, but with the engineer, with the mediated transformation with Agrobacterium, these are highly rooted culture able to produce the artemisinin. This is the mechanisms or how these Agrobacterium rhizogens can induce the root formation, and finally following by the secondary metabolite alterations. So if you infect your plants into Agrobacterium rhizogenis, and then your plant will give the signal, plant signal to the Agrobacterium rhizogenis genomic, what we call the virulence the regions here. So this is Vir A and Vir G, this operant in the TI plasmid in the Agrobacterium. And then they make the complex between Vir D1, the others operants, and then the immature complex. And together with the other complex, the operant can goes to the plant cells. So enter the plant cells, cytoplasm here. And then those two, there are two genes which integrated into the cytoplasm of the plants. And then with integration of two genes can stimulate the root formations. So therefore, you can stimulate the root formation, genetically root formation with the Agrobacterium. In the same time, it can also stimulate the production of secondary metabolites related for the wild types. So we can also control whether the genetic transformation that's happened already in our transform. So normally we do in with the PCR, there are three marker gene, what we call role A and role C. Role A and role C, those two genes which are responsible to stimulate the root formation in the plant. And then Vir D2, this is Vir D2, not role D. Vir D2, normally doesn't have any effect in the formation, root formation. Therefore, if you check it in the your transform highly rooted culture, that you can find two genes, role A and role C, in your plants come from the Agrobacterium rhizogenis. So you cannot detect these two genes in untransformed roots. So and then we also check it here. You look at it here. This is just simple TLC analysis about the from the leaf. You look at this the white leaf. Yeah, the white leaf, the artemisinin here. The white leaf is contain artemisinin as well, but the untransformed root doesn't have any artemisinin. So we could provide any artemisinin in high root culture. So we also applied this technique also for the acetococyte. Yeah, the acetococyte also can be improved with the from acetococyte hydrolysate culture about almost 2 times, and we also can produce the aryl naphthalene lignin, just histidine B. This is anti-cancer lignin from the callus. Yeah, from the callus we only have 2 milligram per gram dry weight. But the transform mediated agrobacterial mediated transformation, we are able to enhance the production 5 times, right? About around 10.8 milligram per grams. That has been published in Journal of Natural Product. So this is technique, what we call actually combinatorial biosynthesis. The technique has been developed to discover new compounds or to enhance the production of secondary metabolites which is low content in plants. The general concept is combination of biosynthetic pathway from different organisms genetically. So we took, for example, we take the gene from the plants and we can transform to the microorganism and let microorganism produce our secondary metabolites. This is very nice technology right now has been developed. So an organism serves precursor, for example, primary and secondary metabolites to be metabolized into secondary metabolite by expression of foreign gene. So we should transform our foreign gene into the host, and then the gene can be expressed into protein. The enzyme can convert your metabolite. So this is our roadmap actually. Yeah, we have for 5 genes. Yeah, in the next slide, I would like to explain to you about the biosynthetic pathway of artemisinin formation. So we have farnesyl phosphate scientists here, either from morphadian scientists, cytochrome P450, double bond reductase, aldehyde dehydrogenase. We clone it and we put in E. coli. We transform in E. coli. We transform in yeast and Bacillus subtilis as well, and we do genetic modified, and finally we feed it. We fed it and we analyze the artemisinin and derivative compounds. So we analyze the production of this compound in this recombinant microorganisms. And the other way, we also put it again in the artemisinin because the lower contents we stimulate again. We put it, we engineer the gene and we put it again in Artemisia annua plants, and we do also genetic engineering. Finally, we fed it, and then we can have the artemisinin. So this is the roadmap. So why we do artemisinin? Because artemisinin is very important anti-malaria right now. It's tierce line therapy. Yeah, what we call, because most of anti-malarial agents have been resistant for the Plasmodium falciparum, right? The parasite can cause the malaria. So therefore, the WHO since 2004 recommended for what we call artemisinin-based combination therapy. But the use of artemisinin is very huge use of artemisinin. So therefore, we need the sources of artemisinin that can available for the production of anti-malarial drugs. In Indonesian cases, since 2002, 2020, 10 years, you think ACT, artemisinin-based combination therapy, can reduce about 33% of malaria cases. So that's mean this compound is very interesting. So we have this seedling. We have several kind of the culture for this one. This is a structure. So this is a very complete the biosynthetic pathway. They are from the metabolic pathway into the MEP pathway. So that we started with the 5 key genes actually, which are responsible for the biosynthetic pathway. What we call farnesyl phosphate scientists, from IPP to the MAPP. We have farnesyl phosphate, and then you have other morphadian scientists here, and then you have cytochrome here that catalyze 2-step and the double bond reductase and aldehyde, and there is some spontaneously step to produce the artemisinin. So what we do, we clone all those 4 genes. But we have the other gene here, but I just chose 5 genes into several microorganisms. I would like to explain to you with the E. coli and Saccharomyces cerevisiae in this kind of presentations. So this is what is the way, the strategy to do that one. Yeah, you look at this one, right? This is strategy to clone those genes. We have FPS here, ADS. This is biosynthetic pathway. We took this gene. Actually, we can do synthetic gene. Sometimes we also isolated gene from the plant as well. But most of them we just order the synthetic gene and we plot it here. This is farnesyl phosphate, the fragment farnesyl phosphate. This is what we call cloning factors, expression factor. We also have expression factor here, cytochrome and IDS. We put together and we put all five actually, and we have one more, the gene that can be put in a factor here. So we have recombinant plasmid that that can be transferred into E. coli. So after transformed to the E. coli, we analyze the enzymes expressed in E. coli. So you look at here, there are some, if you compare with the wild type here, so we analyze all gene expressed as the protein in the E. coli. So we also analyze. This very simple analysis with the TLC. We do analysis as well with GCMS and HPLC just to show you with the recombinant, the recombinant E. coli. Able to produce the new, actually new compound that has been found previously in the, E. coli. So that's mean those compounds produce it because of transformations. And we hope we are analyzing this compound. We hope, of course, those compounds are artemisinin or might be the derivative of artemisinin, for example, aldehyde, artemisinic aldehyde, artemisinic acid, for example. So that's for in E. coli. So we also construct 6 genes, actually 5 genes from the key gene, and we have one more gene that can regulate all 5 genes in the Saccharomyces cerevisiae. Into this is the yeast. The yeast is also eukaryote compounds. What is the strategy to do that one? So you have your gene here. This is from pigeon TEZ. This is cloning factor. You have FPS. And then you cut it, and your P-BAFI, this is factor for special for this Saccharomyces cerevisiae. You cut it as well with the same restriction enzymes, and you combine them together. So and then you can insert to the Saccharomyces cerevisiae here, right? So this is one gene. The next step is we also cut it again for ADS, for amorphidine synthase. And you put it here as well, right? So maybe, and then finally you will have a 6 genes. You look at this one. This is the homologous recombination based cloning strategy, and you have all 6 genes in this plasmid, and you transform to the E. coli. So this is the analyze. After that one, we analyze by PCR whether our gene already transformed into E. coli. So we already confirmed that all 6 genes have been successfully transformed in Saccharomyces cerevisiae here. This is ADS. You look at here the ADS, and all 6 genes have been transformed successfully, and we analyze with the PCR. So we also analyze the protein expression. Here you do this ADS here, only ADS, put it here. Some others, some of them cannot be expressed very well, so we do optimizations. So this is again the TLC analysis. We found this one. This is the artemisinic acid, right? Just look because the time is very limiting, so I can just do very short. This is artemisinin. Those other lines was were our recombinant with and without induction. So you look at this one, right? This is the transform 6, which contain all genes can produce the new compounds related for the artemisinic acid here. With after inductions with the galactose. Galactose, 2% galactose, so we are able to analyze the production of this one. That's mean our Saccharomyces cerevisiae is able to produce the artemisinin derivative because of transformation analysis here. So this is also the SPLC analysis. SPLC analysis. This is artemisinic acid compound. So this is our transform full. This is also we overlay this one. Several compound have been produced with our recombinant Saccharomyces cerevisiae. Yes, right. So this is also nice publications published by Miyahisa. We have also published it just in the beginning of this year in the Advanced Pharmaceutical Bulletin. Maybe you can also look at this. It's free or open access journal, the progress of our research in the artemisinin again in yeast. So we are also preparing some article again with the 6 gene now. We hope our analysis for the recombinant E. coli can confirm us that our recombinant E. coli and also Saccharomyces cerevisiae already able to produce the derivative of artemisinin. So this is the other example. Yeah, what have been published. This is production of flavonoid. Normally, flavonoid never been found in E. coli, right? I think it's our knowledge, and E. coli never produced flavonoids. But the flavonoids can be made as the host for production of flavonoid. This is example. This is a bioflavonoid biosynthesis of started from phenylpropanoid here. This is phenylalanine. This enzyme phenylalanine ammonia lyase here, SCCL gene, chalcone synthetase here. And then you have chalcone isomerase. This is 4 gene, catalyze the phenylalanine into phenosembrin, and also catalyze tyrosine into naringenin with the 4 steps, right? And then Miyahisa construct 4 gene in one plasmid. And there are 2 other ones, flavon 3 hydroxylase, flavon synthetase as well, and this is flavon synthetase here. So this is the construct actually for the people who are working with the genetic engineering. We are familiar with the picture, maybe just to introduce for the natural product scientists. So this is just a picture to show you the plasmid. This is 4 genes transformed into the PET factor, expression factor. This is 2 gene regulation gene, and this is 2 genes what in the middle and at above in the brains. And then we have also 1 more gene. Miyahisa also 1 gene, and all this gene transform into E. coli. Yeah, and once you succeed to transform those gene into E. coli, and then the gene can be integrated to the genomic, DNA genomic of E. coli. So the life is still started. Yeah, the DNA can be replicated and can be transcribed into RNA and can be produced into protein. And those E. coli, recombinant E. coli, able to produce the protein which are responsible to catalyze the flavonoid production. So what is this nice picture here? So you look at here, right? In the below one, this is wild types. We know that there is no E. coli which produce flavonoids, of course. So nothing here. It's PLC analysis, right? And the SP analysis of flavonoid produced by engineer E. coli after getting the gene from the plant. You look at this one, right? With the 4 gene combination, Miyahisa found E. coli, the 3 compounds, what we call creatine here, right? Creatine number 3, after feeding with the phenylalanine, because phenylalanine is the precursor, and the D4 is apigenin. Apigenin is the other compound with using the tyrosine as the precursor. So we have also 1 here, the phenocembrin, and also naringenin. Yeah, that is a very nice result. So that they got actually, and then the nice way that if you are able to produce this compound from the E. coli, so you need only to just produce it whatever you want. So for the plant, you need for example, few months, for example, right, or even year. But in E. coli, you just grow it overnight, and then you can also scale up production, maybe 1 liter, for example, and you get just isolated flavonoid or maybe artemisinin here. That is very nice technology that can be applied to produce the very low content of secondary metabolite from plant. And again, with the other E. coli recombinant, they have several recombinant E. coli. So this is also the other HPLC, the chromatogram, so that they are able to produce galangin. Yeah, galangin is intermediate. I mean the end product, the end product of the flavonoid. What I already told you the previous slide, the biosynthetic flavonoid, and they also can have chimeferol here. Chimeferol, right, after added precursor into the E. coli. but the E. coli doesn't have any alanine, right? Doesn't have tyrosine as well. So you need to feed it, to feed the E. coli as well. So this is very nice. Maybe my time is almost finished. How many minutes more, Professor Sarunsan?
Yes, you are having 3 minutes more.
Oh, 3 minutes more. Okay, thank you very much. I will go so fast. Okay, so this is the other compounds, right? So again, I told you that wild type doesn't have any flavonoid. So this technique have been applied for several important compounds. Normally we choose the commercial drug, and they actually that originally derived from the plants, but with lower contents, but synthetically chemistry is not able to produce it. So we do this technique, of course. We do, maybe we can also, let's say for Taxol, right? Taxol also the terpenoid compound as well. So what I can conclude here, that our research successfully transformed the gene from antimalarial artemisinin into plants. We use also the plant as well, but I didn't present it here. E. coli, what I tell you, Saccharomyces cerevisiae, and alter this country metabolite profile and host microorganisms. E. coli and is, however, the target compound should be analyzed more detail. And then we would like to tell you that the enhancement of secondary metabolite from plants could be done using genetic engineering. So synthetic biology, this is the technique. Combinatorial biosynthesis allow us to produce secondary metabolite from plant in microorganisms. So this technique have been already applied also not only from the plant, but also from the protein therapeutic from human, erythropoietin. Erythropoietin is the protein therapeutic from the blood, from the human blood, but we are not able to make the drug from human blood. So they take the protein, the sequence of protein, they transform into E. coli and lets E. coli to produce this one. This technology platform has been already commercialized, and the product from this synthetic biology have been already in the market. So I think that's all my presentation, but I would like to share this one. I'm happy to have to join with this ASOMPS 17, Professor Altman from in Malaysia, and also Professor Intan, Professor Nohadliani, and I would like to invite you for the next ASOMPS. Yeah, next ASOMPS. Actually, we hope this pandemic COVID-19 can finish soon, so we can do it offline. Actually, Malaysian also planned to do it in online last year, right? Because of COVID, they cannot postpone it, but they cannot postpone it again, so they do it in online. Hopefully, for the next ASOMPS, it will be organized in Bandung. I think we come back to the Bandung, and I think in 80s we organized in 90s. I think Professor Samsul Arifin Ahmad organized it in Bandung as well, in Padang, and now we come next year. Hopefully, we come to the Bandung. Yes, let's join and share our research finding here. This is an announcement. So this is our some of our group here, the very young, very enthusiastic group, very optimistic group, to do this experiment. So I would like to thank all of the people who are contributed in this research. So of course, I would like to thank for your attention. Thank you very much for your attention.
Thank you very much, Prof. Elfami. Very, very interesting talk. Yeah. Okay, I would like to open for questions, perhaps from the participants here, if there are any. At the moment, there are none in the chat box. Or if I may start, Prof. Elfami, I just have a general question. How do you think that in terms of commercialization by to produce artemisinin using this approach, how can it be commercially cost effective compared to large plantation of the plant source and extract the compounds?
Yeah. Okay, this is the good issues, right? Always the nice issue, right? For commercializations, is it economically or not? So actually, if you are able to produce in E. coli or maybe in the yeast, I think the 2014 or 2013, the group from America, already able to produce the hydroactimicinic acid from yeast, right? About 45 milligram per liters culture. So I think because you can scale up it easily, and then with the culture, because for the seed, you only need it to keep it in -80, for example, in glycerol stock. And if you, whenever you want to produce it, and you just took it very small, for example, small instrumentation, yeah, and you put in the colony, and then you can scale up it in very short times. I think it's more economically. Some have been already in a commercial way, yeah. What I think what you should invest is just a medium, right? A medium.
Yes,
you
yes.
can also engineer your medium as well.
Okay, thank you for the answer. Yes, yes. So we have one question here from the Slido chat box. Doctor, from your studies, is it possible to use E. coli and create another painkiller from plants that can replace penicillin?
Oh yeah, that can be the technique, yes. But you need to analyze very well. For example, you have to know the biosynthetic pathway of penicillin, and you already know the sequence of the gene, for example. And you can also just do order the synthetic gene of this biosynthetic pathway, and you can clone it. Yeah, you can clone it, and you put an E. coli. So in E. coli. So if you are succeed, and maybe you can produce this penicillin using E. coli. What I told you that erythropoietin, actually, erythropoietin is protein, right? So protein should be easy because it's not metabolite, not metabolite. The protein, if the E. coli able to produce the protein, that's it. Yeah, you don't need to analyze the metabolite. Yeah, this is a protein, so it will be able. So right now we have also the very recent technology, what we call CRISPR study, right? CRISPR and biotechnology that can be applied. Yeah, can be applied for this cation. And I believe that I mean, development of science and technology will give more update in this technology, then we can apply for production of very low for very low secondary metabolites, even from plant as well. Whatever the activity, yeah, painkiller, anti-diabetic, cancer, for example, we can produce it in the E. coli using the synthetic biology, combinatorial biosynthesis. Thank you very much.
All right, thank you. Okay, is there any more question? I think we are right on time for your presentation, yeah, Prof. El-Fahmi. Thank you. So over to you, Prof. Shamsun.
Yes, thank you very much. Professor Rosanna, I think you were going to introduce the next speaker.
Okay, all right, thank you. So
thank you very much.
I think we would like to flash the e-certificate. Maybe I will, yeah. Okay, so we would like to thank Prof. El-Fahmi for his present talk. Thank you very much.
Yeah.
Okay, so next, we are very blessed to have Professor Dr. Edrada Ebel, okay, to join us, yeah, in this session. Professor Edrada Ebel received her doctorate in natural sciences in pharmaceutical biology from Julius Maximilians Universität Würzburg, Germany. She did her postdoctoral fellowship in the Department of Chemistry, University of California, Santa Cruz, and was a senior research fellow at the Institute of Pharmaceutical Biology and Biotechnology, Heinrich Heine Universität Düsseldorf, Germany. Professor Edrada Ebel is currently a senior lecturer, associate professor at the Strathclyde Institute of Pharmacy and Biomedical Sciences, University of Strathclyde, the UK, and heads the Natural Products Metabolomics Group at the Strathclyde Institute of Pharmacy and Biomedical Sciences, and is currently author of more than 140 publications and 4 patents on marine natural products. Her expertise comprises both natural products isolation, structural validation with modern spectroscopic techniques. Professor Edrada Ebel has won awards and international fellowships on the application of metabolomics on natural products research. Her lab is amongst the pioneers in establishing the application of metabolomics for targeted analysis of bioactivity secondary metabolites. So with that, I would like to invite Prof. Edrada Ebel to present her talk entitled Using Concatenated NMR and MS Analytical Data for Metabolomic Guided Production and Isolation of Bioactive Natural Products. The floor is yours.
Thank you very much, Professor Rosanna Othman, for the nice, kind introduction. So I would share my screen. All right. So am I loud and clear?
Yes, very loud
Yes.
and clear.
Okay, thank you very much. So first of all, I would like to thank the organizing committee, Professor Hadiani, Professor Jalifa as well, for inviting me to the 17th ASOMP meeting to present our work at the University of Strathclyde. Actually, I think the first ASOP meeting I've attended, I was still a student, and that was in Bukit Tinggi. I can't even remember the year right now. Okay, so for this afternoon's talk, I would like to introduce the use of fused data or concatenated analytical data from two types of platform as to be applied on metabolomic guided production and targeted isolation of bioactive natural products. So my team is part of a multidisciplinary consortium in Europe. So for example, we usually get our samples from Greece, and this is a group from Crete, as well as Iceland, but as well as around the shorelines or coastline of Scotland. So for example, we have collaboration with Heriot-Watt University, SAMS, as well as SMEs in Scotland, like marine biopolymers. We also do collaborate with SMEs from around Europe, for example, on our bioactive testings, like Pharmac in Norway, as well as Axum in Italy. And regarding our analytical methods, for example, algorithms and programs, we closely collaborate with VTT. And here with VTT, we were able to, for example, flexibly use such softwares like MZMind. Okay. However, for this talk today, I would be focusing on more international collaborations we have even outside of Europe. So in addition to Iceland, we've got collaboration with Sao Paulo in Brazil, and of course in Malaysia. So I will be looking at the talk together with, I think, with UKM and Terengganu. Okay. So why metabolomics? So we use metabolomics as a tool for drug discovery. In the literature, they are usually coined as well as secondary metabolomics other than natural products metabolomics. So here in these type of metabolomics work, we embed macros using natural products databases to enable us to efficiently do rapid replication. In addition, or in parallel to that, we use multivariate analysis software to investigate patterns in our data. So which means we are using metabolomics as a selection tool, for example, in plant and microbial strain, to be able to determine chemical novelty. Second, we also use it as a selection to optimize our process conditions, and this is important in integrating our metabolomics data with process variable data. One example is, of course, linking chemistry with fermentation. Third, I think this is the most common one or application we use in our group, is to investigate the bioactivity. So which means statistically, we are able to significantly compare chemical profiles of bioactive and inactive extracts. And lastly, we could also integrate our spectral data with genomic data, and this could confirm the expression of biosynthetic gene clusters, including cryptic genes, or we also call it silent genes, and we could further investigate new biosynthetic pathways. Oh, I hope my screen didn't freeze. Okay, so we know as a natural products researcher, looking for our target metabolites or our bioactive compounds could be a maze system. Yeah. However, we did find out that metabolomic tools could provide clear and well signposted map to the target destination. And we think, what's happening? Sorry, I cannot go to the next one. We think that this is better than our road and traffic signs. So one example is this traffic sign here. This is found down south where you have a roundabout, and in these roundabouts are more roundabouts. So quite confusing, but this I think is a more confusing signage. So which arrow do we follow? Or keep right or keep left? So like in metabolomic research, we need to be aware of road surprises. But most importantly, we don't want to beat this sign wrong way and go back. So I think as a natural product researcher, we need to be efficient and we don't want to waste our time and resources. So we do follow this workflow, okay, to manage the hurdles of the replication through metabolomics. So we start with isolation and culturing, particularly if we're working with microorganisms, then goes to extraction. And our extraction procedure is similar if you're dealing with plants as well. Okay, and then we do metabolomics analysis using mass spectrometry and NMR. Parallel to this, we do our taxonomic identification and genome mining data. And
sorry, Prof.
all together, yes?
Your slide is not moving, or you're still on the same slide?
I'm still on the same slide. Oh,
okay.
Which slide do you see at the moment? Is it
the maze with the dot?
Oh, all right. I think I'm already on the next slide. Okay. So how do I do that?
Maybe you unshare and share, right?
Okay. Oh, that's technology, right? So yeah, could you see the right slide?
Friend, you can be enemy. Yeah. Okay.
I know.
Now we can see. Yes.
Okay. So right. So yeah, so this is the workflow that we usually follow. So as I've said, we start with isolation and culturing. So culturing is, of course, when we are working with microorganisms. And then once we have the cultures, then we go into extraction. The extraction procedure we follow is the same as what we do with plants. Okay, once we have the extract, they are subjected to metabolomics analysis. And the platform we're using is both mass spectrometry and NMR. Okay, so once we've got that, then this is in parallel done with taxonomic identification as well as genome mining data. But to bind everything, since we are only interested in bioactive compounds, then we also need to do bioassay screening. So all these data are very important in our metabolomics approach. Okay, next slide. I don't know, it seems to be hanging. Oh my God. Cannot go to the next slide. I think I'll have to share it again. Okay, just bear with me. I don't know what's happening with technology at the moment. So I'll stop the sharing. I'm not moving on the next slide.
What if you don't do the slideshow?
I don't do what?
The slideshow. You don't go into slideshow. You go into the.
I don't go to slideshow. Okay. won't let me. Oh my God. Okay, I'll stop. How do I minimize this?
Or can you send me through WhatsApp your slide? I can try project here
Right.
in the group that you have.
Well, I don't have WhatsApp. Okay, now I've
Okay,
got
sorry.
this. All right, let's see. I don't have WhatsApp on my
Oh,
computer.
okay.
Right. Oh, I'm really very sorry about this.
Yeah.
So
Okay.
can you see my screen right now?
It's coming. Yeah, we can see it. Yeah.
Okay, so I didn't use it. Okay, good.
Yeah.
So right, so we're finished on this. Okay, so I don't know where I stopped. Right, so I'll go to directly to the analytical platform. So as I've said, we've been using two analytical platforms. One is mass spectrometry and NMR. So there are advantages and disadvantages of these two platforms. So we just take the advantages of each. So for example, in NMR, the generic sample preparation is reproducible. We just use one universal solvent, which is dimethyl sulfoxide. However, the great disadvantage is you could only see high concentration of the compounds that you are analyzing. When it comes to mass spectrometry, okay, there's a focused sample preparation depending on the type of technique we're using. So if you're using GCMS, then you are only limited in using non-polar solvents. However, with electrospray ionization, then you use more polar solvent. So that differences in polarity already plays a very important role in the analysis. But the great advantage of mass spectrometry is that you could see low concentration of components, especially if you're dealing with low yielding potent compounds or target metabolites. So this is our predefined metabolomic workflow. Okay, so we always start with mass spectrometry. Sorry, now it's getting, and we're using both positive and negative mode because not all compounds were ionized in the same mode, which means if you're doing an untargeted analysis, then you have a picture of both type of compounds. And once we have this data set, it's transferred to processing, and we usually use MSETmine in collaboration with Finland. And this will do peaking, the convolution, the isotoping, alignment, and as well as formula prediction. So the formula prediction is very important for us because we could use this data for the replication. Sometimes we also do MSMS analysis because then it's easier to do a structure elucidation of your target metabolites. So in the replication here, we start to embed our data set into the databases. We also do a data cleanup here. For example, we working with microorganisms, we need to get rid of all our beta pigs. Once we've got this, we also get then the results from our bioassay and NMR data, put them into a multivariate analysis. And we do use SIMCA and MetaboAnalyst. We also use our package to generate our heat maps. In here, we already pinpoint our target metabolites or biomarkers. And once we have these biomarkers, then we start the discovery of our novel metabolites, structure elucidate them using, again, NMR and mass spectrometry. So for these talk for today, I will be dealing with 3 application projects. First one is optimizing the production of fish antiparasitics from Algoriphagus marincola. Second is assessing the seasonality of Brissonemia intermedia from the Brazilian Cerrado flora. And lastly, comparing Malaysian and New Zealand honey. So the first project, which was done by a PhD student from Portugal, Dr. Bella Sanchez. So a brief background is that we received this anti-parasitic active extract from 7 days of cultures of Algoriphagus marincola. So this is geothermal bacteria isolated from Iceland. So they were inoculating it by single colony, and that affords a very low biomass. So our team was to scale up these microorganisms. However, to scale it up, we could only use 10% inoculum. However, we were losing the anti-parasitic activity in 7 days. So our hypothesis is to fuse the data sets, which will validate the production of the bioactive compounds, which we think is occurring at an earlier growth phase. So our objective is to improve the sample clustering and group classification. So the main question is, can we validate the optimization method during scale up on the production of these fish anti-parasitic isobranch alpha and beta hydroxyl fatty acids if we directly fuse the data from 2 analytical methods? So the aim is to increase the yields of these anti-parasitic compounds. So very briefly on our methodology, so we have actually 2 blocks. So block 1 is our NMR chemical shift, and block 2 is our mass spectral data. So we put them together, we scale each of the block using either Pareto or univariate. So for those of you who are using statistics in their research, so Pareto, the great advantage of using Pareto scaling is we are able to see the low intensity ion peaks or chemical shift peaks, which is very good in determining low intensity potent biomarkers. However, in univariate, we use this less because it tends to average all the intensity of the peaks, which means we lose this low yielding components. So if you want a more detailed description of this methodology, you could find this in our publication and scientific reports on the metabolomic analysis of Tithonia diversifolia. So going back to the project itself, so we have here the blue as the 10% inoculum and the purple spots as the single colony. So we could see already changes between day 1 to day 7. So day 1, the extract still seems to be similar to each other, but on day 7, you could see here a big difference between the metabolites. And that explains why we were losing the bioactivity on day 7 on our 10% inoculum cultures. On the NMR, there's also a trend between day 1 to day 7. However, here there's a similarity between the cultures at around day 3. Okay. And when we concatenate the data, day 1 to day 7, then we could also assume the similarity in the cultures between the 10% inoculum and the single inoculum from day 3. When we did the log transformation of our data, okay, we could see here a very nice trend of the single colony from day 1 to day 7. However, on the 10% inoculum, we could already see our targets beginning to be produced on day 3. However, we lose the yield on day 7, and that explains again why we are losing the bioactivity on day 7. So as a conclusion in these projects, that it's very good that we are already producing our target metabolites at day 3. So if you're considering an industrial scale-up, we don't need a 7 days fermentation scale and stop the fermentation at day 3. We already have achieve our optimum yield. Second, looking at the reproducibility of our replicates on mass spectrum, we could see here that our replicates are coming into different type of clusters. On the NMR, they're still under one cluster, but we do see differences between the replicates. However, after fusing our data, we could see that the error has been corrected. So we all have the replicates coming together in one cluster. Okay, so that is actually, we say, oh, the great advantage of using the data sets, because it does correct all these errors. Going to the second project, so this is the Brazilian project. So we look at Brisonema intermedia from Brazilian Cerrado, and this was done by a PhD student, of course, from Brazil. So her name is Ana. So a brief background. So Brisonema intermedia, the infusions of the barks and the leaves is used by the public in Brazil because of their antiseptic, antimicrobial, anti-hemorrhagic, and anti-inflammatory properties. However, Anna is more interested in looking at the anti-inflammatory properties in this case. However, the inspection of the MS and NMR spectral data between the samples collected at various seasons did not really indicate any clear differences in terms of the biomarker components. So her hypothesis was to look at the seasonality, which is actually the major environmental factors that exert the influence over the synthesis and accumulation of certain secondary metabolites in Brysonema. And she's more interested in looking at the anti-inflammatory triterpenes. So the main objective is to pinpoint these seasonal biomarkers from unique perceptible constituents, particularly for the anti-inflammatory triterpenes. So to show you, so here is a principal component analysis of the unfused data. So we have here mass spectrometry and NMR, and you can see that the data sets or the samples collected between the various seasons are actually overlapping. Although here we could see that the summer samples is a little bit separating from the rest of the samples. However, in the NMR, our problem was that the summer samples was not really conclusive in terms of its separation because it was overlapping with the autumn samples. Here we had a better separation of the spring samples and the winter samples. So after concatenating the data, we had a clear separation of the summer samples, and then we have a sloping trend from winter to spring in the production of the secondary metabolites. So we got interested in looking into the summer samples because we think that the triterpenes are being produced during this season. So we use a PLSDA so to separate the summer samples and define these biomarkers. If you look at the NMR data, we could see that there is highly aliphatic chemical shifts between 0 to 5 ppm. And the molecular weight is between 400 to 500 Daltons. So on the next slide, you're looking into the replication, then we could see from the summer samples that we have a higher pentacyclic triterpene content in the summer and triglycosylated flavonoids in the spring. So that could then be further validated statistically using this box bars. So we could see here the green boxes representing the triterpene contents in the summer season. While here we could see the yellow boxes, okay, we have an increase in the relative abundance of the triglycosylated flavonoids during the spring season. Okay, so to conclude this project, so the seasonal factors affecting the metabolite production, particularly of the triterpenes in the summer, is we've got higher temperature, which is above 25 degrees centigrade, where we get a solar radiation of between 2000 to 2500. And there's also lower humidity, which is below 70%. And the rainfall is only between 0.15 to 0.10 millimeters. And lastly, we go to the third project, which is comparing Malaysian and New Zealand honey. So brief background, the Malaysian honey exhibited bioactivity against the breast cancer cell line, while the New Zealand honey was found inactive. So inspection of the MS and NMR spectral data between samples do not indicate any apparent difference between the Malaysian and New Zealand honey. So this was quite a problem. So the hypothesis is that the fused data sets will improve the clustering and correct the differences between the replicates to define the bioactive biomarkers in the Malaysian honey. So the objective is to pinpoint these low-yielding bioactive biomarkers from more perceptible components. So here is before the fusion. So there is already an obvious separation between the Malaysian honey, which is represented by these green dots, and the blue dots by the New Zealand honey. However, we could also observe some crossovers between samples from New Zealand and from Malaysia. In the mass spectral data, the difference is more obvious. So we got a very good separation between the Malaysian and the New Zealand honey. So looking closely on the differences in the features, we could see here, for example, on the right upper quadrant, which represents the New Zealand honey, we have these blue dots, which really doesn't occur in the Malaysian honey. And this blue dot represents low chemical shifts or upper field chemical shift, which means we have a higher lipid content on the New Zealand honey. While in the Malaysian honey, where we have more green dots, that is actually between 5 to 6 ppm, and those represents like phenolic chemical shifts, so which means there is a high phenolic content on the Malaysian honey. If you look at the mass spectral data, so we have here darker blue spots, which represents molecular weights between 100 to 200, while here we have the lighter blue, which is a higher molecular weight compounds in comparison to the New Zealand honey. But when we did fusion, okay, the differences became more obvious. Okay, so here we've got the Malaysian honey, and here the New Zealand honey. But interestingly, there is a clustering occurring between the two samples or two sets of samples from New Zealand. So here we have the North Islands and the South Islands. But to summarize first, the result between differentiating Malaysian from New Zealand honey, the New Zealand honey has higher lipid content, and this is represented by the higher density of darker blue dots, while the higher density of the lighter green spots indicated lower molecular weight compounds. So which means the New Zealand honey had lower molecular weight lipid content. Going back to the Malaysian honey, we had higher sugar and polyphenolic content represented by higher density of lighter blue dots, while the higher density of yellow-green to orange spots, okay, which you see down here, indicate the higher molecular weight compounds. So in simple terms, the Malaysian honey had this glycosidic phenolic compounds with higher molecular weight, which could be responsible for the anti-cancer activity against breast cancer cell lines.
Prof. Abel, you have 4 more minutes.
Okay, yes, I will tackle it. Thank you very much. So here, looking again, the difference between the North and the South Islands. So the North Islands is richer in acetylated compound, and that could be explained by the signals between 2 to 3 ppm, while the Southern Island has higher phenolic content. So to summarize, okay, we found out that the fused data sets validated the optimized production and harvest method for the target metabolites. Second, our fused data sets also improved the linearity of the production and the presence of the metabolites against the respective parameters considered in the respective studies. And lastly, the fused data results also had better clustering and corrected the differences between replicates to define the biomarkers or the target components. So lastly, of course, I would like to thank my group. I think some of them, or one of them is in the audience at the moment, Dr. Nurkhalida Kamal, right? Our collaborations with different universities, and of course, a collaboration with SMEs around Europe, particularly with Pharmac, and that is the anti-parasitic project. And most important is our funding agency in the UK as well as in the European Union. But most important at the moment, so I would like to thank you for your kind attention, and I would like to apologize regarding the humps when it comes to the technology. I'm sorry about that. Okay, so now we are open for questions.
Prof. Shamsun, you might need to unmute.
Okay, thank you very much, Professor Edraba Abel, for your very nice presentation, and it's very informative. So this session is now open for question. Still now there is no question, but I have a question. And I'm very much interested regarding your projects of honey, and you compare the Malaysian honeys and other countries' honey. So I have a very I'm very curious to know that sources of honey. So for example, most of the cases honey are based on different floral the different floral connection. So there are mustard honey, there are lychees honey. So it varies season to season, the collection and quantity. So have you ever could have some time to focus that whether the constituents or metabolites are coming from the flower and how that influences the production of honey, and will it be kind of that so for example mustard honey, this will have definitely specific class of the compound. So if you kindly enlighten me on it. Thank you very much.
Yeah, thank you very much, Professor Shamsun. Yeah, that's very true. So when my student was looking at the type of flora that was found in New Zealand, okay, as well as in Malaysia, of course, the differences is already very obvious. The flora, of course, where it was collected, really reflected the type of compounds or metabolites the different honeys were producing. Yeah. So yes, it is very dependent on the geographical flora. This honey are these metabolites are being
so another thing
represented.
is another related question is that how can the biomarkers can be varied from flower to flower, like
Oh, yes. So that's quite difficult because I think because, for example, in a region in Malaysia where the honeys were collected, okay, it's quite diverse. So which means it's not near in a, unlike if you consider, for example, a plantation, and then you have a honey, then you would know that these honey will only be collecting these type of metabolites. Okay. Yes. However, I think according to Yusnaini, okay, who worked on this project, was that this was like forest honey. So which means there is really a diversity of the type of flowers or plants that was around it. The same thing with New Zealand. I think that was like a natural forest as well. But we could, of course, see a very obvious difference between the South Island and the North Island. Yeah. Okay. So I think the North Island, if I'm not mistaken, is the one which is more tropical, and the South Island is more temperate as well. Temperate. Yeah.
So, thank you, Professor Abul. There is a question that from the viewers. Could the temperature of certain country influence the lipid content in honey?
Yes, yeah. Maybe it's not really the temperature, but as we've already discussed, is that the type of plants that's being produced. Yeah. So, for example, the North Island, I'm not really sure now which one is the more temperate region and the more tropical region, whether it's the North Island or the Southern Island. So at the moment, I really cannot make a conclusion if the North Island is the more temperate. Okay. Sorry, it's the more tropical type of temperature, then why is it producing more lipid content? So we could not make that generalization when it comes to New Zealand honey, yeah, because it's, I think, the South Island, which is more the temperate type region, okay, is producing higher phenolic content, yeah, which is more similar to the ones being produced by the Malaysian honey. However, the Malaysian honey is glycosidic in nature, so which means the sugars are attached still on the phenolic structure, while those coming from New Zealand, they don't have the glycosidic structures. Yeah.
Thank you very much, Professor Abul. Now, now we are over Professor Rosanna. Thank you.
Yeah, Professor Shamsun, actually, there's one more question. It's in the WhatsApp actually. So it's from Dr. Intan for Prof. Abul. So she asked, Prof. Wan, can we balance between sensitivity of the analytical platform with the scaling? For example, mass with UV and NMR with Pareto to decrease too much data to deal with. Unless, of course, we want to consider the most out of all peaks or signals.
Yes, you could do that. I think yeah, my student from Brazil, I think, tried to play around that between the Pareto and the univariate. The only problem is that when you're fusing the data, you have to either, you could use both, that you use, for example, only univariate for NMR and then Pareto for mass spectrometry. But I think the advantage is that if you already know your target metabolite, okay, and you know the signals where it's coming in, and you know that they are of higher intensity, then I think univariate method is a better scaling method. Yeah. So I think she did that as well when she was looking at the triterpenes using the summer samples, where the triterpene samples were just shooting up of the spectrum, both in the NMR and the mass spec. So I think she changed it as well to univariate.
Okay, all right. So is that Prof. Samson? We are done with the questions, right?
Yes. Thank you very much, Professor Idraba Abel.
You're welcome.
Yes. Thank you.
Okay, thank you very much for the very interesting lecture. Next, okay, last speaker of the day, but not less important. We are happy and we have a pleasure to welcome Professor Mylene Hui to join us. Okay, I can see Prof. Hui is together with us. Okay, but she has kindly provided a recorded video of her presentation because of internet issues. So before that, let me introduce Prof. Mylene Hui. Prof. Hui is a professor at the chemistry department of the Mindanao State University, Iligan Institute of Technology, the Philippines. She is the program leader of the Natural Products and Drug Development Research Group of the Premier Research Institute of Science and Mathematics of MSU IIT, and presently is director. As director of PRISM, she has established and leads its core program, the Discovery, Development and Delivery of Natural Products. She obtained her Doctor of Science degree in Biological Sciences and Bioorganic Chemistry from Hiroshima University, Japan in 2015 as a Mombukaga Kusho Scholar. Prof. Hui has authored and co-authored more than 50 international papers and has presented in a number of international conferences and symposia. She was the 2016 Novartis Next Generation Scientist from the Philippines and the 2015 National Research Council of the Philippines Achievement Awardee in the field of chemical sciences. Currently, Prof. Hui is implementing 2 research programs being funded by the Department of Science and Technology, Philippine Council for Health Research and Development, as well as the Philippine Institute for Traditional and Alternative Healthcare. With that, I shall present the recorded presentation by Prof. Hui.
I cannot hear the voice, Professor Othman. Do you have any problem?
Please share again and share with sound. Can you increase the
The
volume?
sound needs to be turned up.
Yes. Sound is
No sound, yeah? Let me
sound is very faint. Yeah, it's
Faint.
not loud. Yeah, very faint sound.
try again.
Could you perhaps stop it and start again if the
Yeah.
sound can be turned up?
Okay, let me try again. Checking. Do you see the video?
Yes, it is visible.
Okay, let me try.
We still cannot hear the sound. Okay, it's okay for Professor Rana. Let me do the sharing. I have the video.
Prof. Roseanne,
Yes.
let me share the recorded video from my end.
Okay.
Maybe it would be better. I just hope.
Yes. Okay. Sorry about that. I don't know what happened. I've tried it. Okay. All right.
It's all right.
Yeah. Okay. Good.
All right.
All yours.
Yeah. Let's see if I can play it.
Yeah, I think still the same. The volume.
So the volume is still the same.
Yeah, I cannot hear well.
Okay.
All right. Let me just present it. Let's see if
Okay,
I can do it.
Yeah, can. By all means.
Yeah. All right. Thank you. Let me see. Stop share. Well, this is technology. Where is that? Right. Slideshow. And, okay. And all right. So, can you see my slides now?
Yes, it is visible.
All right. Thank you so much.
Very beautiful.
I hope my sound is all right.
Yes,
Okay.
Yes, loud and clear.
All right, thank you for the kind introduction. Good afternoon, everyone. I express my deepest gratitude to the organizers of ASOMPS 2021, with special mention to my friend, Professor Zu Zakaria, for this opportunity to share with you my research group's journey in exploring the rich biodiversity of Mindanao, Philippines for natural products. The Philippines is regarded as one of the 17 megadiverse countries in the world, since it is blessed with an exceptionally rich diversity of species and an amazing abundance of endemic species. Among the megadiverse countries, the Philippines, next to Ecuador, is the second smallest. The Philippine archipelago is composed of 7,107 islands, with a total land area of 30 million hectares and covers only 2% of the total land area of the Earth. Yet 52,177 described species have been recorded here, comprising 2/3 of the Earth's biodiversity and between 70% and 80% of the world's plant and animal species. The country occupies a world ranking of 4th in terms of bird endemism and 5th in terms of mammalian and reptilian endemism. Of the estimated 7,620 species of plants documented in the country, 5,832 are believed to be endemic. This puts the Philippines in the 23rd ranking in the world and the 6th ranking in Southeast Asia in terms of plant diversity. In addition, the Philippines is the center of the Coral Triangle, since its marine resources are made up not just of coral reefs, but of seagrass beds, mangrove and beach forests, fisheries, invertebrates, seaweeds, marine mammals, and many others. This is according to the 4th National Report to the Convention on Biological Diversity in 2009. As such, the country is rich in both terrestrial and marine biodiversities. In Mindanao, in particular, these biodiversities have remained undertapped and underexplored. Now, about Mindanao, let me share some facts. Mindanao is the 2nd largest island in the Philippines, in the southern part of the archipelago, surrounded by Bohol, Philippine, Celebes, and Sulu Seas. It is irregularly shaped, and it is marked by peninsulas and heavily indented by the Davao and Moro Gulfs in the south and by the Iligan Bay in the north. Mindanao is often referred to as the land of promise, and rightfully so. It is an island of limitless potential, home to a vast array of lush, rugged, faulted mountains, forests, and seas. Volcanoes occur in many areas, including Mount Apo, which is an active volcano in the southern part of the Central Highlands and is the highest peak in the Philippines. Mindanao has narrow coastal plains and broad, fertile basins and extensive swamps formed by 2 river systems. The rare and famous Philippine eagle is also found in Mindanao. Now about my university, the Mindanao State University Iligan Institute of Technology, which is located in Iligan City, is one of the 11 satellite campuses of the Mindanao State University system and is the flagship campus for science and technology. It is dedicated to academic excellence with a commitment to the holistic development of the individual and the society. It was founded in 1968, and it is known in the Philippines for its excellence in science and technology and its passion for extensive research and community involvement. To realize our vision, mission, and goals, we dedicate ourselves to our code of conduct of accountability, commitment, excellence, transparency, integrity, teamwork, and participation. Now, on its 53rd year, my university has come a long way from its original mandate and is looking to reach new heights of excellence. It is rated as a 3-star university by the QS, ranked 3rd among 6 Philippine universities by the Green Metric World Universities ranking, and it is ranked 6th in the Philippines by the Webometrics ranking web of universities. Thus, in Mindanao, my university is in the forefront of instruction, research, innovation, and community involvement. In terms of natural products research, the university has evolved from mere ethnobotanical surveys and phytochemical screenings of medicinal plants in the 1990s to the early 2000s to serious drug discovery and development work beginning in 2012. This growth was primarily catalyzed by 2 things. The first was in 2012, when the university was designated as the country's first Tuklas Lunas Development Center. With this designation, we have been able to secure funding for research projects which have enabled us to procure basic laboratory equipment essential to the conduct of natural products research. The second impetus was the establishment of the 5-story research building, the premier research institute of science and mathematics in 2012, which became operational in 2017, with the subsequent formation of the Natural Products and Drug Discovery Research Group, which focuses on the discovery of bioactive extracts and compounds from terrestrial and marine sources of Mindanao, and their development into herbal preparations and new drug leads for various diseases, with emphasis on cancer. These 2 catalysts have enabled us to elevate our research to include the conduct of biochemical assays, cell-based assays, as well as bioassay-guided isolation and purification studies. Our research mandate was given another thrust with the establishment of our core program, the discovery, development, and delivery of natural products, which targets to produce pharmaceutical leads, dietary supplements, nutraceuticals, and cosmeceuticals, among others. I'm sorry, it's raining again heavily. I hope that I'm still being heard. The research programs or projects that are being undertaken by our research group follows two parallel paths of drug discovery. The first track is the herbal track with standardized products as outputs, and the drug track with lead drug targeting candidates ready for preclinical studies as our outputs. So within this context, our group has been implementing several research projects since 2012, including drug discovery and development from Mindanao plant species, the traditional medicinal plants utilized by indigenous peoples of Mindanao, discovery and development of natural products from Mindanao marine sources, utilization of endemic spices for the development of various natural products, as well as lead optimization, molecular docking, and determination of admitox properties of active compounds. In the interest of time, I can only share with you selected highlights of some of these projects. So the first big collaborative research work we did was in 2012 to 2015 on 18 medicinal plants found in Mindanao, including some of which are traditionally used by one indigenous people of Mindanao. For this project, 90 organic and aqueous extracts were prepared and subjected to preliminary screening using standard protocols pertaining to their toxicity, antioxidant properties, and antimicrobial activities. So principal component analysis was done to visualize the relationship between the antioxidant capacity of the plant extracts and their chemical composition. Data was reduced to two principal components, which accounted for a total of 73.891% of the variance in the data. In Table 1, we see that PC1 showed the highest loadings for phenolics, DPPH, and total antioxidant capacity. PC2, on the other hand, revealed contributions from phenolics and DPPH. Meanwhile, FTC assay had low loading for PC1, but showed a negative correlation to PC2. Moreover, if we look at the loading plot, it shows that the TPC and DPPH are positively correlated. This result is in line with several studies on different plants, fruits, and vegetables, which have reported positive correlation between phenolic compounds and DPPH anti-radical scavenging activity. However, TPC did not show significant correlation to TAC and is not correlated either to FTC activity. Thus, for the plant samples in this study, the phenolic content contributed only to the DPPH anti-radical scavenging activity and had insignificant contribution to the total antioxidant capacity and lipid peroxidation activities. Metabolites, on the other hand, or other than phenolics, might have contributed to the TAC and FTC activities shown by the plant extracts. The PCA score plot, as shown, exhibits that the plant extracts are widely spread over the graph area. So there is no clear distinction in antioxidant activity in terms of the type of extraction. It can also be seen that the DPPH, the total phenolics, the total antioxidant capacity, and the ferric thiocyanate assay results are higher in the plant extracts on the right side of the plot than those on the left. So among all the plant extracts, however, the ethanol extracts of Metroxylum sagu, the decoction of Prunus grisea, and the aqueous extracts of Eucalyptus deglupta and Itlingera elatior exhibited the highest contribution to the first component, PC1, indicating that they have the highest antioxidant activity in terms of phenolics, DPPH, TAC, and lipid peroxidation inhibition. Thus, these results imply that these 4 plants have the potential to be developed further as herbal supplements. Select active plant extracts of considerable weights were further investigated by some of our graduate students who work in the lab. And one of the plants was Ageratum conisoides, which is commonly called goat weed. And it is traditionally used in the Philippines to treat various ailments. The chloroform soluble partition gave the highest antioxidant activity than the other extracts in terms of total phenolics content and % inhibition of lipid peroxidation. The purification of this extract led to the isolation of several known compounds, including coumarin, sinencytine, and 5-methoxynobilietin, which have established biological activities which can actually give scientific basis for the plant's many folkloric uses. Another plant which underwent further study is Eucalyptus declucta bloom. It is a huge evergreen tree that belongs to the Myrtaceae family, found in the lowland primary forest in Mindanao, hence its common name, Mindanao gum. It is traditionally used for treatment of asthma, bronchitis, lung infections, and malaria, and also used as antiseptic, deodorant, and insect repellent. Purification of its active chloroform extract gave the U-global IA epimers mixture, which have been found to possess several biological properties which may give credence to the plant's medicinal uses. Equisetum dibili, a fern alley under the family Equisetaceae, commonly called horsetail or scouring rush, was also further examined. Ethnobotanical reports have shown that its traditional uses include treatment for hepatitis, diarrhea, kidney infections, eye inflammation, fever, liver problems, bone dislocations, and rheumatism. Results of the antioxidant activity evaluation of its various extracts show that the chloroform soluble fraction consistently gave the most remarkable results in terms of GPPH, radical scavenging activity, total antioxidant capacity, total phenolics, and inhibition of lipid peroxidation. One of the compounds obtained from the chromatographic purification of the active extract is herniarine, which has been reported to demonstrate antidermatophytic, antifungal, and anti-cancer activities. Our group also further scrutinized a very common plant among Southeast Asian countries, Annona muricata, a small tree that belongs to the family Annonaceae that is popularly called soursop. In the Philippines, its traditional uses include using its leaves and as a compression for inflammation and swollen feet, or also for eczema and skin eruptions, while a decoction of its leaf is used as a treatment for head lice and bed bugs. Also traditionally used as sudorific, antispasmodic, emetic, antiscorbutic, and astringent. 2 of its extracts we found to exhibit high toxicity towards human colon cancer cell line, HCT116, and interestingly, the purification of its active hexane extract led to the isolation and identification of the 3 ficaprinols, ficaprinol 10, 11, and 12. Ficaprinols are free polyisoprenols which have no clearly identified functions in biological systems, despite accumulating at high levels within certain organisms. It is believed that these compounds actually play a distinct role in cellular physiology. Prunus griseia is a member of the Rosaceae family, said to be found in Malaysia, the Philippines, Singapore, and Taiwan. In the Philippines, and the Subanen indigenous people of Mindanao call it as tanga-tanga because they use its sap to treat scorpion bites. There is very little information regarding this plant. However, because the indigenous group recognized it as having curative properties, we included it in our investigation. The antioxidant properties of the various extracts of Prunus grisea were determined, in which the decoction consistently gave the most remarkable results. Purification afforded a mixture of the isomers ursulic acid and oleanic acid. These two acids are commonly and widely found in food, medicinal herbs, and other plants, and have been shown to have a broad spectrum of activities, such that we find these acids already commercially available anywhere else as supplements. Our group also studied a small flowering tree native to the Philippines, Ficus nota, which is commonly called as a sucking tree. It is traditionally used for the treatment of many ailments like fever, muscle pain, UTI, hypertension, diabetes, among others. We found that the crude chloroform soluble portion from the ethanolic extract of the air-dried leaves of this plant has antibacterial properties against Bacillus subtilis and Staphylococcus aureus. From this bioactive fraction, we isolated two new C11 terpenes with an octahydrobenzofuran skeleton, which we designated as Ficus nota diol and Ficus nota non. This is the first reports of octahydrobenzofuran analogs of this kind isolated from this species and other Ficus species found in the Philippines or in other countries. These compounds are postulated to arise from the degradation of carotenoids, just like several C11 terpenes isolated from plants and marine organisms. Patents, we have applied patents for these compounds and their potential uses. From the same bioactive chloroform soluble portion, we further isolated 6,1,4-diaryl butanoids, designated as Ficus notins A to F. Ficus notins A to C are previously undescribed compounds, while Ficus notins D to F are known compounds previously obtained as synthetic derivatives, but have never been found as naturally occurring compounds. These compounds are considered plant-derived 1,4-diaryl butanoids, which are postulated to be synthesized biogenetically from phenylpropanoyl coenzyme A, such as p-coumaroyl coenzyme A. Since 2016, our research has been more focused on work related to identification and characterization of high-value purified active compounds derived from Mindanao marine sponges, with potential use as anti-cancer drug templates. So for phase 1 of the project, 35 sponges were collected off the coast of various parts of Mindanao, and polar and non-polar extracts were prepared for each to give us a total of 70 extracts. These extracts were then subjected to cytotoxicity screening using isogenic colon cancer cell lines at the University of Florida in the laboratory of our collaborator, Dr. Hendrik Luch. The results show that 15 sponge extracts exhibited considerable potential to inhibit the proliferation of HCT116 cells at 110 millimolar concentrations. Following the protocol of the Philippine government's 2-class LUNAS program, which adopts a rational drug discovery and development, wherein hepatotoxicity and nephrotoxicity is conducted early on, these 15 sponge extracts subsequently underwent this test. The results indicated that only 5 extracts passed the criteria and are prioritized for purification. These are the polar extract of Axinella species and the non-polar extracts of Myxella crucifera, Haliclonus species, Onotoa amphiastra, and Tylexocladus species. Now, interestingly, according to the World Register of Marine Species, M. crucifera is only found in the Philippines, and luckily in Mindanao, and literature search revealed no pharmaceutical studies on it yet. As our research lab was able to acquire more cancer cells in the succeeding years, we also tested the priority sponge extracts against the proliferation of mammalian breast human hepatocellular carcinoma, and lung cancer cell lines. The results indicated that among the 5 sponge extracts, AN20P and AN21NP exhibited remarkable activity against HepG2, with AN21NP also showing considerable anti-proliferative potential against breast and lung cancer cells. And so MTT assay guided purification of the active non-polar extract of M. crucifera through various chromatographic means has led to date the isolation of 7-dehydrocholesterol peroxide, scalardecine B, and beta cholesterol. 7-dehydrocholesterol peroxide indicated varying activities against 4 cancer cell lines, which actually parallels its reported in vitro anti-cancer activities against several cancer cells. Scalardecine B, on the other hand, also gave remarkable inhibition of both colon and lung cancer cell proliferation. It is a sester terpenoid, which is among the most common in sponges, while beta-cytosterol has been shown to exert a chemopreventive effect in DMH-induced experimental colon carcinogenesis. It has also been shown to be an effective apoptosis-promoting agent against breast cancer and a potential anti-metastatic agent against lung cancer cells. The priority sponge extract from Axinella species, which is AN20, has been undergoing purification through chromatographic processes. So far, 3 active, albeit known compounds have been isolated from it. These are the mixture of phyllopholactones H and I, which at 30 micromolar concentration exhibited a considerable anti-proliferative activity against the liver carcinoma cell HepG2, and the other compound is scallarine, which at the same concentration actually only mildly inhibited proliferation of HepG2 cells. Now, based on literature, phyllopholactones H and I are known secondary metabolites of the marine sponges belonging to the genus Phyllospongia. On the other hand, scallarine-based sesquiterpenes have been found to display a variety of pharmacological activities like cytotoxicity, anti-tubercular, antimicrobial, anti-inflammatory, among others. Our work on the purification of this active and other active samples of the top 5 phase 1 sponge extracts is still in progress. Yes, okay, I'm wrapping up. While we continue to work on the, yes? Can I continue? I have a few more, 3 more slides. Okay, thank you. While we continue to work on the putative priority phase 1 sponge extracts, we are also doing parallel work on our phase 2 samples, which involves an additional 50 sponge samples collected from the coast of Iligan Bay. So we again prepared polar and non-polar extracts from the phase 2, screened at 30 millimolar for anti-proliferative and pro-apoptotic activities against colon carcinoma cells and lung carcinoma cells. So to determine which among these will be prioritized for further investigation, we adopted the tiering classification established by the University of the Philippines for the Tuklas Lunas program. So the results indicated that we have 11 extracts as top tier against ACT and 3 extracts as top tier against A549. For the pro-apoptotic activity, we have 19 extracts as top tier against HCT 116 and 9 extracts as top tier against A549. Now, these top tier extracts are therefore prioritized for further investigation. Now, interestingly, if we look at HCT 116, there are 5 common extracts with top tier classification for both anti-proliferation and pro-apoptosis. So these extracts will be further investigated for their bioactivities, conformation through secondary and orthogonal assays. Also, we will be doing hepatotoxicity and nephrotoxicity profiling before we proceed to purification. Now, cancer is a complex disease, which involves various features like uncontrolled proliferation, resistance to apoptosis, metastatic potential, cell migration, among others. Thus, it is logical and practical to cover all possible routes of action test samples may have against cancer. So currently, this project is also screening the phase 2 sponge extracts with the third primary assay, the scratch wound assay for migration inhibition studies. And so our research work would not have been possible if not for the support of our university, our funding agencies, our collaborators, our students, and research staff. To them, we are forever indebted. And so with that, I end this presentation. Thank you very much for your kind attention.
Thank you very much, Professor Mylene. Very nice presentation, and as well as it was very informative. So the session is open for questions. So we can see a question here. So the question is that whether you could see screen compounds at 30 millimolar or 30 micromolar. This is the question.
Come again, sorry, I was reading the chat.
So could you screen the compounds against some biological targets within the concentration of 30 millimolar or 30 micromolar?
Oh,
because the concentration of the compounds.
That's 30 micromolar.
Yes,
30
exactly.
ppm. Yeah.
30 ppm. Okay.
Yes. Sorry about that.
Okay. So
I think Professor Mary has a question in the chat box asking for who did. Yeah.
Your sponges were beautiful, the photography and the taxonomy. So I just wondered who did the taxonomy. Did you do that, or are you in collaboration with somebody?
We are in collaboration with a professor at the Biological Sciences Department here for the phase 1. But for the phase 2, we are still to have them identified. So we are actually looking for a collaborator for their ID.
Okay, well, it's a great start to name them. So the scalorane compounds, of course, you don't have a gem dimethyl group. You have an extra methyl group, so that you know, that's worth pointing out, that they're homocestoterpenes rather than cestoterpenes.
Yes.
Because
all right.
you've got a methyl and ethyl group rather than a gem dimethyl group.
Thank you. Thank you so much for that input, Prof. Mary. We will correct our report on that.
No, don't correct it. It's just it's worth pointing out because it's an unusual biosynthetic feature.
Yeah, okay. Thank you so much.
It's not a regular cestoterpene.
Yeah. Thank you.
Hello, Sakawa, question. I have a question. Hello. Do you have any,
yes, please, Professor. Please carry on. Yes.
Do you have any nitrogen or the halogen containing substances in your marine natural products?
So far, those are still the compounds that we have isolated. Our work is still in progress, and we hope to get interesting compounds. We hope to isolate interesting compounds, those containing nitrogen or halogen containing compounds, hopefully with our active extracts.
Thank you. Thank you.
Thank you.
Yeah,
Professor Mylene,
sorry, can I just make a comment? I mean,
yes, please.
The NMR spectra are being run in the States, so you have to send your compounds overseas. So it makes it quite hard because you don't actually know if you've got a pure compound or not. You're kind of dependent on that, on getting that NMR data. And of course, in the current COVID situation with lack of planes and so on, that must really complicate the research to not have that capacity. I know, of course, that an NMR machine is going to be installed in Iloilo City. So there'll be another one in the Philippines. And so, you know, maybe hopefully that can allow you to do things to do the NMR in country rather than having to constantly send it overseas, because you
Yes.
always have a question mark about whether the compounds are going to survive that journey
Yes.
overseas. So
Yes, actually we
congratulations
are.
on what you do against that background.
Yeah, actually, we are quite happy with the decision of the government to procure another NMR machine, which will be put in Iloilo in the University of San Agustin,
Yep.
because they told us that this will cater the Mindanao and the Visayas universities. So
Yep.
we're looking forward to less complex, NMR measurements, because as you said, it really, with the pandemic, it's making it more difficult for us to send and wait for the results from abroad, from our
Yeah,
collaborators.
no, I'm really pleased to hear that you're going to collaborate with them on that, in that regard. It's critical, and just all the best for that.
Yes, thank you.
Yes, Professor Mylene, I have a very simple question. So how did you conduct the nephrotoxicity test? Nephrotoxicity.
For the phase 1, we submitted it to the University of the Philippines, because at that time we didn't have yet the capacity to do them. But for the phase 2, for this year, we are going to establish this type of test here in my lab. So hopefully we can do it, and we will be having a training with the University of the Philippines team for this assay. So they're using kidney cells for the nephrotoxicity.
Okay, so it was on through the cell lines. Okay, and
yes, cell lines
could you have the chance to test your cysteine type of compounds against helminthisis, any kind of helminthis, because that you mentioned it, that is it was a goat feed, and that was the traditional uses for the anti-helminthin as an agent.
Yeah, not yet. We haven't done so, because we need the synthetic group to do the production of that compound. We cannot rely, and we cannot. It's not also sustainable to collect the plants again and do the isolation again. So we rely on the synthetic group. We have one here to produce the compound synthetically, and then do some more tests.
Okay, that's very nice. Thank you.
Thank you.
So,
okay.
Yes, Prof. Rosanna. Yes.
Okay,
So
great. So I think we have reached the end of today's program. Okay, so the research in natural product is so vast. We would like to thank Prof. Uy as the last presenter of the day, and especially to all presenters. Indeed, the presentations today have, by all the eminent speakers, have underlined the theme of the symposium, contextualizing natural product science for societal relevance. Yeah, we would like to thank all speakers for their valuable share of knowledge and research accomplishments. I hope everyone has benefited immensely from the presentations today, and we hope to meet everyone again tomorrow. Sorry for any shortcomings from our side. Yeah, this technology can sometimes be very challenging, especially when we are, halfway across the globe. Yeah, but it is very interesting and a very nice experience. Yeah, to share with our younger generations later on. Okay, so we hope that to see everyone again. And
Prof. Rosanna, sorry, I need to.
Maybe we should have a photo.
Yes. That's what I'm going to ask.
There are a few questions recently arising. So
yes,
the questions, all the screening compounds are known compounds. So this is question from one of the participants. All the screening compounds are known compounds. This is a question.
Yeah, I think what he means was all the ones that we have isolated are known compounds. Yes. So far, and but from Ficus Nota, we were able to get novel compounds.
Okay. Thank you. Next question is, after the screening, what do you do for those bioactive extracts? Will there be developed into some pharmaceutical products or pharmaceutical natural products for pharmaceutical users, basically? So this is the drug development procedures, whether you are taking these compounds to the pharmaceutical industries.
Yeah, well, it will depend really actually for the plants, for plant samples, we hope to come up with herbal supplements. But let's see. But for those coming from the marine, of course, it will have to be developed as drug templates if they have promise.
Okay. There is a last question. And so this question is that, please share your plan on proceeding to discover potential bioactive metabolites from the traditional plants.
Thank you. Terima kasih. Thank you very much. I'm very sorry, Asakawa. My face photo are blocked by some technological errors, so I'm very sorry. Okay, so tomorrow probably you can see my face.
Oh, okay.
Thank you.
So it's a pity.
Ari-san, thank you.
Thank you.
Thank you, Professor. Arigato gozaimasu.
どうもどうもどうも。ではさようなら。Goodbye.
Good night. Oyasumi nasai.
おやすみ。
I think I have to say goodbye too. It's time to get something to eat. It's dinner time.
Yes, yes.
Right, right.
Oh, we do that.
It's 8
Maybe
p.m.
we can
It's dinner
take
time,
picture.
right?
Yeah.
Yes,
Yeah.
Prof.
Because not also
Thank you.
edible in Japan, okay? Because no, that you presented. That is
Yes.
very tasty. Fruits is very tasty. Yes.
Yes.
Bye bye.
Okay,
Bye.
see you. Arigato.
See you.
Thank you all.
Arigato.
Thank you. Lovely.
Okay.
Bye.
So
Yes, Prof. Rosanna, please.
yeah, so okay, so before we go off, can we have all the speakers to switch on the camera so that we can take group photo?
Mhm.
Prof. Gasson has left.
Prof. Mary has left.
Oh dear.
I
Prof. Elfami, can you switch on your camera? Okay, so Professor Elfami, are you still around? Okay, I think we'll just smile, whoever is on camera now. Okay, okay, 1, 2, 3. Let me just paste this and keep this. Oh dear, it's a pity Prof. Gasson has left. All right, okay, with that, I thank you everyone. See you tomorrow. Goodbye.
see you tomorrow. Goodbye. Take care.
Take care.
Bye-bye.
Thank you.
Bye-bye.
Bye.